Hello. Hello. Hello.
Yes, sir.
Hello.
Hello.
Yes.
Yes, sir. We are just launching. Just a second, sir.
Yeah. One comment I have.
Sir?
Not a scientist myself. If you would involve the unfortunate 18th cross Malaysia, there is one person there, two people were there, one died, only one left. This whole thing.
Sir, everybody listening. What do you want to say?
I'm simply saying what Panditji said. Narendra Modi is saying. Involving the India's quintessential academic institutions, which was a failure, it's a wrong, would have been a better way of doing it, isn't it? It's okay, it's too late now. I don't know whether you are hearing me at all. Can you hear me? Can you hear me?
I hear you. Madam, switch off.
I will go outside.
No. You can microphone. Hello. No, madam. Yeah. Hello. Sir, your face is not appearing. You have to adjust your screen. Okay. Fine.
So somebody is Vishnu Prasad.
But we are not hearing his sound, madam.
He has to unmute. How to share his screen?
You don't understand. He should unmute.
Can I share the screen?
Sir, can you hear me?
I can hear you barely.
Okay. No problem. My voice not important.
வரேன் இருங்க ஒரு நிமிஷம்.
Yes.
There's going to be quite a bit of problem here. 1 second. Let me share the screen.
Yeah. Did you enable, madam? Share screen?
Sir.
Yeah, it's here.
But we are not.
Why Vishnu Prasad is joining now? We can wait.
I don't know. Yes, sir. Who was?
Sir, I will do my best, but with my very poor IQ, we are going to face some difficulties in transmission. It happens. 10% of the calls to India don't mature. But that's okay, we'll try.
We are doing all over the world, sir. What is the problem? Never
How
heard.
can you say what Sam Petroda said about 2 months ago? What he said, Reno, I have done my job. You go to hell, jump out. India has to spend more money, and they're going to do it.
Sir, I don't want to talk politics on a public media, sir. It's very dangerous for me.
Even making comments in a
It's
democratic
dangerous, sir. Why do we unnecessarily comment in foreign soil, sir? No need. We do science, that's all, sir. No.
Just a moment. I'm struggling to get the screen.
But admit, don't admit anyone, madam,
Yes, sir.
because it's a problem.
Some network issue also
Where?
here. It's again connecting.
Here. For us, no.
For mine only, it's not connecting.
Can you see the screen?
No. Sir, you have to go to share screen.
That's what I did.
You go to share screen and click on the screen which you want to present. Madam, you have
I
shared.
just check, sir. It's not. It came now.
One second. This is going to be a little bit of embarrassment.
How do you check whether he's sharing the screen share? Did you enable already, madam?
Yes, sir. I just only host.
You can do that.
Sure.
Enable screen share. Sir, if you want, you can send by email or PowerPoint. I can share it.
That's going to take a long time. We should have done it earlier. That's okay. It's too late now. Probably it is late.
No, sir.
There are also some proprietary reasons why I cannot do that.
We have 15 minutes, sir. No problem. Only 2 minutes to take time. You send the mail. I will share it.
Sir, there are several of them. There are some proprietary reasons why we cannot transmit it.
I can able to.
No, sir. Only one participant.
No, you cannot access this system as much as I cannot access your system. Let me see what I can do here.
Hello.
Enable panna solunga madam.
ಸರ್ ಇದು ಲೈವ್ ಬಂದಿದೀವಿ ಇದರಲ್ಲಿ ಅವ್ರದ್ದು ಸ್ಕ್ರೀನ್ ಶೇರ್ ಮಾಡಬೇಕು ಸರ್ ಮಾಡಿದ್ದೀನಿ ಬಟ್ ಅವ್ರೇನೋ ಶೇರ್ ಮಾಡ್ತಾ ಇದಾರೆ ಡಿಸ್ಪ್ಲೇ ಆಗ್ತಾ ಇದೆ
Disabled.
ಸರಿ
Yeah, sir, you can do it, sir. You go to share screen, sir.
Mm-hmm.
And you go to whatever the file, advanced or files. You just click in, sir, which file. You go to files. Then you can share. Yeah, it is enabled.
Yes, sir. If he is in co-host, it will enable automatically.
You put it a co-host.
Co-host.
So you are the co-host, sir. You can enable. Sir, can you able to do, sir?
I'm not struggling to do that. I don't know why it doesn't let me do it. It should, isn't it?
Sir, you go down, sir, share screen.
No,
I
no,
know
I will.
that. That's what I'm doing.
No, no, we have to make a co-host. Who is, what is his name? How it is recognized here? Dr. Renu?
Renu.
Okay. So I think now being a co-host, you can go to share screen. Sir, can you go to share screen?
Yes, that's what I'm doing.
When the moment you click on share screen, you find all the folders which is opened. You have to keep the file open.
Suna, come. Can you put the AC, Suna?
Or you can share the file, sir. We will share,
There
upload
are
it.
several of them, sir. There are also some proprietary reasons why we are not doing it.
Okay, okay, fine.
As I explained to earlier. Just a moment. Share screen. I can see your face, but that is not helping us.
Yeah. If you click on the share screen, the green button,
Yes.
you
Sir,
find
click on the share screen. You will see on the top basic, advanced, and files. Then you click on what you like, then you share it.
now it's coming.
Yeah, it's coming.
Yeah, but I need to get rid of this one. One second.
No, we are able to see you and your screen. No problem.
So may I put it on recording
Yes, sir.
after 5 minutes, and YouTube is on.
Yes, sir. It's on.
Okay. Yes, wonderful, sir. It's coming now. So that YouTube also plays on the Facebook.
Can you see the screen now?
Yes,
Yeah, it's very clear.
very clear.
I will do my best, sir.
It is coming out wonderful, sir.
Good. Let us wait until 7:30. The point I was trying to make is, assume we are democratic countries now, recently. Involve the academic institutions. Indian Institute of Science is a bad place, I know that. But still, my counterparts there
Sir, do not record, sir, please.
No, it's not recording.
don't record now. If
No,
you are
it's not.
recording, let me know ahead of time. Okay. If you are going to record it, you are welcome, but let me know so that, because the FTC requires it.
We don't record, sir, anything.
Okay, fine.
I don't want any trouble.
Maybe another way of doing this. One second.
Ramesh, sir, you can present Dr. Renu.
No,
Just-
actually I'm outside.
7:30. Let us go on time.
It is not comfortable here to do that.
One
Oh.
second.
You know, all little 18% GST.
That one insurance.
By the way, one Infosys guy can transform the whole thing.
No, it is too much, ma. Why 1 crore? 5 lakhs.
Hello, Jeff. Hello.
Yes, professor. Yes, professor.
I don't know whether the presentation will go okay, and right now it looks okay. If I were to transfer the files to you and tell you which folder it is, would you be able to display it? Are you on the Zoom?
They didn't allow me actually, so I put in the
Narayanamurthy,
yeah.
I think that is not going to work. Yeah, I can understand. Patla sir, we'll see what we can do. Let us wait until 7:30, right? And also, I agree with you, Dr. Elangovan. We have to keep our national pride intact. This is very important here for every nation, small or big. You will see some contradictions as I move along, if I can move along. Actually, Ramesh, I would like to take a long journey and come back. Are you listening?
Come in, sir.
In my recycling process, I'll become better, I think.
Okay.
Okay, is there audio working? I mean,
Yeah, working. Okay,
very good.
Call me, madam, because I have to tell them.
Ramesh, what is your mobile number?
98450.
Excuse me?
98450.
Just a moment, just in case.
Yeah.
Okay,
10306.
What I get?
let me read it back to you. 98450-10386, right?
306.
Oh, 306.
Yeah,
Sorry.
Indian number.
Yeah, that's okay. 16306.
Sir, we write if you want in chat box, you can read it, sir.
Yeah, I written in the chat box, you can see that.
Yeah, so you don't miss it anything.
It's there in the chat box.
ಎಲ್ಲ including tax ಅಲ್ವಾ? Okay.
How do you admit one more person? He has to register and all that. I don't know.
Yeah, you can admit, sir. You can see in the right side panel, you can admit yourself.
Okay.
Once you log in, you'll get that name on that. Participants. Click on the participants.
This is another person working on the flexible electronics project,
Okay.
competing with Mitsubishi. Yes, Jeff.
Yes, professor.
It is too late. We should have done these things earlier. So I don't think that's going to work. I know you will do a good job on that. I mean, the other people can do it also. It's not that they cannot. There's enough skill. I don't think we are going to go look for computer skill outside India. Right?
Okay.
There are two webinars, one now and one coming up with Europe, followed by Japan. So it's a kind of a clash across Asia and across Europe. And Europe is in the grip of the worst pandemic. I'm not able to get anyone get to normal. Okay, try to join. Okay, if I see your name appearing, I will say admit. Okay.
Yes.
Okay, thank you. Can you? Yeah, I don't think it's a problem in saying, and you can hear me too. We are waiting for 7:30, right?
Yes.
I would like to finish everything within 60 minutes because I don't like prolonged delays,
Yes.
and I have a zero tolerance on delay.
Thank you.
Karoshi,
Okay.
Japan, work to death.
Yeah.
I wish I had all of them in one single file. So this is going to be another issue. I'll get to the other ones. And I got about 3 or 4 of them. I want to conclude on the vaccine development, what's going on at this moment, at this hour here.
Ramesh sir, can we continue? Can we start the meeting, sir? It's 5
Yeah,
o'clock.
I think it's 7:30 here, going to be.
Yeah, you can start now.
Can we start, sir? Or?
Yes, yes, yeah.
Hello, good evening. Welcome to webinar of Pride Energy and Environment Research Institute, Bangalore. It is a great honor and prestige to present Dr. Professor V. Renugopalakrishnan, a renowned American biophysicist. Renu and his group at the Children's Hospital, Harvard Medical School, and Northeastern University currently focuses on the interface between protein engineering and nanotechnology. Professor Renu is one of the pioneers of medical nanotechnology and protein structural analyst in the United States. He has been on Harvard faculty since 1984, starting from assistant professor to a professor. In recent years, his laboratory has been targeting proteins as intelligent and innovative biomaterials for energy conversion through solar, fuel cells, very high density data storage, and tagged on to carbon nanotubes, graphene for direct drug delivery sensors. I have associated personally as a research scientist with Dr. Renu. I learned a lot of light sensing proteins, and he is one of the innovative protein structural specialists in the United States, and we learned a lot from the professor. He teaches and is guiding MD PhD program at Walter Bradford Cannon Society, the first year MD students' new pathway in medical education as a part of Harvard MIT Health Science program, and is currently spearheading a campus-wide effort on bio-inspired devices. Most of his research projects are funded by NSF, United States Air Force funding, National Institute of Health. He was Wallace H. Coulter Chair, one of the prestigious chairship, and as a professor at University of Florida, Miami. He had been a visiting professor in Denmark, Taiwan, India, Mexico. He obtained his BSc from Madras University and entered graduate school in the Department of Chemistry, Columbia University, New York, and a Rockefeller University in New York, working with Professor Harold Urey, is a well-known Nobel laureate in chemistry, and Professor Barbara and obtained his PhD in biophysical sciences in the State University of New York, Buffalo, New York. He is the author of more than 250 publications and highly cited worldwide. Also, he written so many books and 2 monographs in 1991 and 2006. And he also wrote few books, textbooks on bio-nanoscience. And he is a mentor and innovator of bio-nanotechnology program in the United States. He was the editor-in-chief, Journal of Bio-Nanoscience, associate editor, Journal of Nanoscience and Nanotechnology, and also so many journals. He's an invited editor and associate editor of reputed journals internationally. His scientific lineage ancestry traces its origin from Professor G.N. Ramachandran, FRS Biochemistry, to Nobel laureate Professor Stanford Moore, Rockefeller University, New York, on experimental studies of protein structural biology, to Professor Dan Wesley, University of Minneapolis, Minnesota. Dr. Renu generated large human resources in the United States and worldwide through PhD programs and masters in biochemical engineering and biomedical sciences and biochemistry. He is well specialized on Raman spectroscopy of proteins, and his major research mostly on biomaterials for drug delivery mechanism, a modern science in the medical sciences. And we are all very proud and honored to hear and invite Dr. Venkadeshan Renu Gopalakrishnan, and he is currently associated with Massachusetts General Brigham Hospital, Harvard Medical School, Boston, and Boston Children's Hospital, and Center for COVID Innovation. Today, his lecture is Race Towards Rapid Home-Based Detection of Coronavirus. I would like to mention, we can add so on, and Dr. Renu is well known in most of the research labs in the world on biotechnology, bio-nanoscience, and biomedical sciences. And I would like to invite you all to hear the most prestigious lecture going to be delivered by Professor Renu. Thank you, Renu. Welcome.
Yeah, thank you very much, Dr. Ilango. I call him by the nickname Maharaja. He has always been like that. And he has been, I have known him since 1997, January, I believe 18th. That when I was in Mexico at the Universidad Nacional de Mexico. And that was a short, supposed to be 1 year and then turned out to be 2 years. What I would like to do is to capture the very essence and spirit of what's going on in this country. Our entry into this field was on the evening at 6 o'clock on the 12th of March, which is a Thursday, 2020. And 1 month prior to that, it's unproven that I might have had asymptomatic COVID because Chinese never told me during my visits. The trouble started in Wuhan. If you go back to the literature, it probably goes back to the somewhat earlier, and nobody knows when it was. Anyway, we started the COVID Innovation Center here. Mass General is a huge hospital with about 19 Nobel laureates to his credit, and Brigham and Women's about 9 or 10. So together, that two hospitals along the Children's Hospital has garnered something like less than 30 directly and indirectly Nobel Prize winners. I want to capture the essence of this, and I will give you a little bit of background where we are located, and some very generic information. And we are starting a new MD PhD program. There will be no more MDs. 5 years from now, 10 years from now, it will be MD PhDs. And that is the direction we are taking. We are retooling, reconfiguring the medical education completely. This lecture is, the last one was in May 2029. You see the big difference here? I've been talking in many places, I didn't list them here. Our mobility has been severely retarded by what was an epidemic, transformed itself into a pandemic. Why is this screen not moving? Oh, maybe here. Yes, our group consists of many divisions: energy, solar, that is at Northeastern University, and biofuel cells, sensors, point of care devices. Then we are probably one of the very few groups in the world on protein-based microprocessors and terabyte, maybe gigabyte memory at this point. And we also have a very embryonic project, a daring project on the transmission of smell with both electronics that has not seen the light of the day, like many things have vanished. Unfortunately, with the death of its founder, Professor Ahmed Bose. We also do a lot of fluorescence imaging, real time, of tissues. We are now working at the organism, tissue, then down to the atomistic level. We have a group on pancreas, artificial kidney, and artificial liver. I'm very proud to say that's a very high level collaboration with a string of universities, including Tsinghua in Beijing, Fudan University, and also the Peking University is a very big project. Should I be admitting people or you'll take care of them? Ilangovan.
Yes, sir.
Can you take care of admitting, because I'm spending some time on that. I would rather go on the.
Yes.
Can you take care of admitting people?
Yeah. They're taking care of, sir.
So we are supported by many laboratories for protein structure, dynamics, protein engineering, now nucleic acid engineering. We just had a paper just about 2 weeks ago, analytical chemistry impacts nearly 7, and molecular cells coming up at impact 23. So our group is, and the flagship of this entire thing is the New England Journal of Medicine, which was mentioned by Joe Biden in his presidential debate last night. And that is jointly, I'm very proud to say jointly with India. So there are many, many things going on. It is all of value to students who are aspiring to become, India has always produced very high quality students and high quality scientists. Unfortunately, it has been difficult to hold them onto the soil. Okay, now having given a small, very brief introduction and the architecture of the group, this is the aerial view of the Harvard Medical School, a very high density of scientists in this place. And we expect one of my students to go to the Stockholm City Hall one day to receive a Nobel in physiology or medicine. We carefully select them, and some of them are teaching me rather than my teaching them. So this is an aerial view. The Children's Hospital is a pediatric hospital, has a #1 rank, has been there for more than 150 years. All these institutions have been around for a very, very long time. This is where my lab is, in the 14th floor of Center for Life Sciences. This is the Brigham and Women's Hospital, which is now has a name, Massachusetts General and Brigham. It's called MGB. The Center for COVID Innovation is located in this building, but none of us have seen these laboratories for a very long time, since on 9th of March, last time I was in my office. They are operating remotely. I hate to say stealth. Now, the current global situation is pretty grim. Globally, as of 2:45 p.m. Central European Standard Time, 22nd of October, Thursday. More than 41 million cases have been confirmed, and the deaths have been 1.128 million, of which about 220,000 is from the United States. I know there's an underreporting of all this for political reasons. We should not get involved in that. So some of these numbers are probably grossly underreported. We don't know, maybe half of it is reported or even less. If you look at the public health spending on health, public health spending as a percentage of the gross domestic product without cost contained has been soaring. Today, United States alone, as of this morning, is $3.65 trillion. That is approximately 1/7 of the pre-COVID GDP. We don't know the post-COVID GDP certainly is going to be much lower. Now, to make room for a pandemic of this magnitude, these figures have to go up. Somewhere the money has to come from. It's not simply money, it's a question of delivery of the healthcare. It's the remotest points in this country, and it is a big challenge in China and India too. China comes next with about 1.2 trillion. India, Ayushman, and all of them put together maybe 300, 400, 500 billion, half a trillion. And all of these are underreported or overreported. Therefore, the healthcare spending as a percentage of GDP has been consistently going up. This figure is outdated. Things have completely changed now. Now, India has many advantages. Number one, there's a strong demand. There are attractive opportunities. There's a rising manpower, especially the younger generation. And the government is doing as much as it could with all the constraints. In all the democratic societies, there are always problems. China remains a major driver of global growth and also the healthcare. As of yesterday, it was over 1.2 trillion. That is still not adjusted to the COVID. But the cooperation between US, China, and India, of course, Japan, and the European powers is there, but not as much as it should be. Together, we can solve the problems better than individually. Unfortunately, there is much to be done in that area. Now, if you come to the entire healthcare cost, molecular diagnostic is approximately 30 to 40%. The first battle, does a person have COVID? This is taking a longer time. There are 278 companies as of 6 p.m. yesterday in the United States alone. And all of them are competing in the market. Some are way ahead, some are in the middle ranks of the ladder, which is quite big. And there are some smaller companies too, very small companies too. Now, if you look at the genetic testing, the blood screening, now we have moved to saliva, especially for children, where multi-inflammatory syndrome is a major problem in pediatrics, affecting the children below 18 or 17. That number is increasing. The top of the bracket above 70 is going up, especially if a person has comorbidity. So the diagnostic market is huge, but a lot of it is wasted, redundant. Redundant tests is adding to the cost of the healthcare in the USA. There's a blood test taken almost every 2 weeks, which is not necessary, and it's not going to help us. It's only going to bloat the cost. Now, we have several consortiums, all of us are funded by the NIH. Our funding runs in millions of dollars. Direct costs, indirect costs, the institutions get an equal amount, even more. One is the bioengineering NIH consortium. Our partners are, number one, Stanford Medical School. We are tied up with them very closely, despite less than 2,800 miles. University of California, Berkeley is where the microfluidics design, fabrication, mass production is occurring. That's headed by Dorian Liebman, he's a professor of bioengineering. Then we have the Boston Children's Hospital. We have the pediatricians and Dr. Gerard Berry, Michael Agus. Michael Agus is right now the busiest man here. He's in the neonatal ICU. I can get in probably for 2 minutes once in a while, if I'm lucky. Then we also have the Brigham and Women's Hospital, the various divisions, especially the immunology and the pulmonary. Our pulmonary division is very important here. The entire Stanford is headed by Ramaswamy Paul Murugan from the Rajapalayam near Madurai. Is from the Madurai Kamaraj University, which has produced very high quality scientists. And he is in the microRNA project for early detection of cancer. So many things are going on here. And the other thing is in the material science part of it, we have a very well-known person of Kerala origin, Pulikal Ajay, Madhava Panikar Ajay from Rice University, and Tarangat Narayanan Nambudri Narayanan at the Tata Institute of Fundamental Research in Hyderabad, Central Electrochemical Research Institute in Karaikuri. It goes on and on and on. They are right now interacting with the 21 groups in India. It's not an easy task. They're working on a New England Journal of Medicine paper with Anthony Fauci. Now, the next consortium is a microRNA prostate carcinoma oncology consortium. It's funded by the National Cancer Institute. And here, what we are doing is we are also collaborating with the North Dakota State University. I think there's one paper on that just about a few months ago in the Journal of Bone and Mineral Research. There is a paper in oncology. There are several papers, paper galore, but I'm trying to ask the students not to look at papers, look at science itself and the patient care. I'm more concerned about the patient care. So we are one of the groups that combines the material science, biochemistry, molecular biology, medical science, clinical care. In fact, I joined the doctors. I don't have an MD. I didn't get an MD because I was roaming around the states of Chennai doing nothing instead of taking up the MD from Harvard. And that would have enabled me to be a full-fledged physician with a strong backup from basic science. Now, so that is the microRNA prostate carcinoma. Prostate carcinoma is becoming serious now, like the colorectal cancer. Some people think the colorectal cancer has been triggered by the COVID. We don't know. There are more folklores on COVID than real stories. When President Trump talks of fake news, you have to include this also. There's a lot of information floating around. We have to sort them through and see what is the most important one. Having said that, let us move forward. There are many biomarkers. How do you characterize the disease? Every disease unleashes a number of antibodies, number of biomarkers. In the case of cancer, it is a microRNA. In the case of COVID, it could be a whole host of biomarkers that are unleashed when the virus begins to attack a host cell and make an entry into it. So for early detection, in the case of COVID, early detection is quite tough. It's a very silent disease that may not even show up. I don't know who has asymptomatic COVID. It's very difficult to assess it. And we're looking at a very large number of monoclonal antibodies, as well as a number of other biomarkers that are unleashed during the infection process. Now, the next one that we have to concern ourselves, how do you assess them? In the case of COVID, it's not one biomarker, it's a multiplicity, a plethora, an abundance of biomarkers. Now, biomarkers crosstalk the inflammation, the entire immunology, the molecular aspects of immunology is currently a very hot topic. I am not an immunologist. Our group has only one immunology link in it, is Dr. Dwayne Wiesman, who's an MD PhD from Harvard and Yale University, and he is working, and we completely depend on him to provide not only monoclonal antibodies required for eventually even for the vaccines. The vaccines there are only 3 companies as of yesterday, 2 or 3 companies. Moderna is one of them. We have reached an agreement with them to supply monoclonal antibodies, and I'll talk more about it as we proceed. So therefore, the whole idea, how do we detect the virus? Do we detect the virus? At the same time, we should also detect the antibodies. Supposing there had been a prior COVID infection, then there should be antibodies in the serum, in the plasma, or in the saliva. They're using saliva for children. So we have a bio-saliva bank now, like a biobank for the blood, which has been around for a very long time. We still don't know what type of blood groups are less immune. For example, O plus is supposedly immune to COVID infection. All these things are still being investigated. Actually, many groups are working on it. The information flow here is every hour. As I'm talking to you, I think there have been 2 papers in the last 20 minutes. That means we have to outsmart them, we have to read them, make sure that we are at the top of top. That's where it starts. Now, Dr. Soumya Vishwanathan is a group leader in the area of glucose sensing. She has some of the highly cited papers. She is an MD, and she is also a medical administrator, and she has played an important role in our research. And she has the glucose, cholesterol, lipid sensing projects, and coming very little into the COVID. Chronic illnesses, we thought 75% of the US healthcare dollars spent on chronic illness, mainly cardiac issues are there. The infectious diseases have come to the forefront now. And I think we are not going to see one pandemic. It's going to be multiple pandemics. The healthcare cost, as I mentioned, is 3.62 trillion, if I am right. That is more than the GDP of France, and slightly more than the pre-COVID GDP of India. All the GDPs are subject to major revisions. Now, proceeding further, the whole principle of detecting the virus is based on the concepts in biosensors. Biosensors, we have sensors everywhere. As we are talking, sensors are watching us. Biosensors are picomolar, 10 power minus 12 to 10 power minus 15, even sub-picomolar to up to femto. We need that level of detection to be able to detect an object that is so small, and we need, it should be rapid, it should be sensitive, it should be cost effective, preferably a throwaway, which one can use in the privacy of the home, in the living room or the drawing room in India, where you can yourself detect, like that. It's very similar to pregnancy test, or measuring a glucose, or measuring the oxygen pressure using an oximeter. We do this routinely. At one time we were not. We are turning the homes into hospitals, mini hospitals, because it's dangerous to go to the hospital, unless it's a crisis, an accident of some kind. Typical sensing, there are many forms of, I'm not going to spend that. 2 things that are very important in the COVID. One is the fluorescence. The second, from our point of view, is the field effect transistor. This is where physics comes in. Then the virus CoV-2, SARS-CoV-2, right? By the way, SARS was preceded by MERS, which is Middle East syndrome, respiratory syndrome. Before that, in around 2004, we had the SARS outbreak in China. I don't know why China had been the major source of these viruses, and they also were taken aback. There are some reports indicating 2019. December is a very sensitive topic to China. I don't talk to my Chinese colleagues on this, and they say, don't go into the history of this. Let us see what we can do to solve it. So there are 2 methods. One is the fluorescence and the field effect. Why fluorescence? An interesting story. The other day, I think on Tuesday, we had 21 institutions participating. We had even some hospitals in Arunachal Pradesh reporting certain rare types of COVID, but the mutations, the genetic diversity of the infection in India is very broad. Where did it come from? Through the land road, apparently on the China-India border. That is one story. It might have come through other sources. People are traveling back and forth, and so there's no way we can. Now, the virus is a very dynamic entity. It undergoes mutations. Why it undergoes mutations? We don't know. In India, certain types of mutations in the spike protein, the S1, I will show you a picture of that. We're going to see a lot of molecular structures here later. I hope there will be enough time. is the mutations occur in the spike protein. The spike protein is the one that makes the entry into the host cell. The mutations are all clustered in the S1. We thought one single mutation of an aspartic acid 6 protein to a glycine is what causes it. Lo and behold, it appears there are other mutations. As I had a long chat with the National Center for Biomedical Genomics in Calcutta. So we are all joining together with the inspiration provided by Dr. Anthony Fauci, provided he can extricate himself from the big mess that he is in right now. He is not in a mess, he's being made into a mess, unfortunately. He is an excellent physician and a scientist. So in India presents a genetic diversity. There is another very sensitive topic where I have to be careful. Those mutants that you see in India seems to be not entirely confirmed with the Afro-American population here. See, it comes at a very wrong time. It can be misused. And I know one person who wants to abuse it. I'm not going to go into the politics part of it. I'm only a scientist. Therefore, fluorescence and the field effect transistors are very sensitive to detect the virus. At the same time, some of the sensors we are developing based on FRET, which is a force of energy transfer on the field effect transistors, we can also detect the antibodies. So from a very, very small droplet of blood, no necessary to do the pricking. We can do it by a nasopharyngeal swab, or we can even do it by saliva in the case of children. We can detect not only the virus at a pico level, at a picomolar level, or even sub-picomolar level, at the same time sense the antibodies too. We've got to have an idea whether this patient had asymptomatic COVID. This is the biggest threat. We don't know how many people have it. There are probably many, many people have it, and it's gone unnoticed, and there's no way of detecting it early enough. Now, moving forward very quickly, we start with the lab and a chip, but scientists have been very poor in commercializing ideas. We are not money oriented. A very distinguished a friend of mine told me that science should be dissociated from money, which I think is not what happens in the free market economy. But that's a different topic. I'm not going to go into that. So the papers in this field are Whitesides in Nature, and Soumya Vishwanathan from our group is a much celebrated paper, never saw the light of the day still, with the Nature Medicine. And some of these journals and the patterns that follow it are very, very important. Today's basic science is tomorrow's technology, right? So that's how we should go. I'm going to skip it. We may run into a time problem. Where does the graphene come in? Graphene is actually a discovery totally unanticipated. It's a platform on which you can attach a molecule. The molecule can interact with the virus. Virus will transfer it to the graphene surface, amplify, you will get a simple electrical radar. Blue means you're okay. Open a bottle of champagne. If it's not blue, it's red, pack up, exit to the nearest ICU, they'll take you, or go to another place. It's up to the ambulatory service to decide where you are going and your luck. Microfluidics is an important part of our research because when you talk of the flow of blood, or even a nasopharyngeal swab, which we do these days, actually the COVID test is very simple now. There's a lot of mumbo jumbo about 3 months ago. Here you can get it done in 20 minutes and the results will come out exactly 24 hours later, which is it doesn't help us. In case of acute COVID, we've got to know the results immediately on the spot. One way of doing it is what we are working on. Just a view of the Harvard Yard. This is one of our. Do you know how I can switch screens? I had to go to the next file. Okay. How do we go to the next file very quickly?
Sir, you can open that file again, then other file which you want to show it, and then again do the same way, share screen, and select that file.
Okay, 1 second. If I can do the previous one, I should be able to do this, right?
Yeah, you should open that file, the next file which you want to.
Okay, that's what I'm going to do now. Yep. Can you see it?
No, not yet, sir.
Okay.
Yeah. No. Yeah, now it's coming.
Okay, very good. That's great. But how do I toggle? Diagnostics and testing. That date is wrong. It's actually 22nd of October. But anyway, how do I? Yes, I think it seems to work. Now, this was borrowed from some of the WHO. There are different types of diagnostic tests available. Some diagnostic tests detect infection and others detect an immune response. Testing plays an important role, but this testing alone cannot take up our time and the resources. So it's got to be very fast. So as I said, there are nearly 278, if I'm not misquoting what I said earlier, companies working on this now. Of which about 10% of them, about 27 of them, are accelerating the speed of research, the pace of research. And some of them are also migrating towards the vaccine. Because supposing we find that a patient has COVID, how do we treat him? Remdesivir was just approved by the FDA, I believe, the last 24 to 48 hours. The cost of it is about more than $3,000. This is another very thorny subject, the cost of these medicines, but I don't want to get into that now. So there's an immune response to COVID, there's a diagnostics test for COVID, and breaking the chains of transmission. We don't know how the transmission occurs. We thought social distancing at 6 feet would be enough. If you look at the paper in Science, hardly 6 days ago, it may not be 6 feet, it may be more. How does it help us? It doesn't help us, it makes it worse. So the diagnostics is one part of it. Some of us are beginning to see whether our hybridoma-derived cell lines, which we developed jointly with the University of Potsdam in Germany and Boston, and we have German collaborators in this, French. We do not collaborate with China on this subject. And sitting at Harvard, there have also been other problems here. So we don't want a knock on the door, midnight knock on the door. So we are trying to avoid any confusion. I'm not even concerned when COVID started in China. I want to keep out of it. Who started it? I mean, it's an infection from bats. The immune response to COVID-19 infection, the whole coronaviruses have 4 structural proteins: spike protein, nucleocapsid, envelope, and membrane protein. During an infection, the body's immune system makes antibodies, immunoglobulins, as you know very well, for a very long time, that bind themselves to the structural proteins and neutralize the virus. The spike protein have come to the forefront, and the nucleocapsid proteins, we now have barely scratched the surface on this. Very first scientific paper on that is being written up and pushing the students, and the students cannot go into the lab as much as they want. The most important antibody in the response are the immunoglobulins. IgM antibodies are produced first during a response. What do the diagnostic tests for COVID detect? The virus. We're after the virus. I call it the devil. It is a beautiful fluorescence. In nighttime, when you look at it stored at minus 80 degrees, it's pretty scary if you're alone in the lab, if you're permitted to enter the lab. Sometimes I think it is telling us, give me a chance, I'll wipe out the entire city of Boston. Don't play with me. Now, leaving aside those fears and our prejudices, as well as many, many things associated with the COVID, we are trying to detect the viral RNA and COVID-19 viral antigen. Immune response is the antibodies. There are several of them, especially antibody is a large protein molecule, about 150 kilodaltons. It has an antigenic domain. We are using monoclonal antibodies using hybridoma cell lines. We are now switched to camel antibodies. They are called camelids. Camelid is shorter, less antigenic than the human. Then we went to llamas. Llamas is not working out well. Then we went to another closely related species called alpaca. Then we are also looking at the shark antibodies now. Therefore, there are two things, detecting the virus and detecting the antibodies. All of them have to be done in one single platform very quickly. Testing the COVID-19 infection, the timing of testing is important. See, once it is there, it's not going to keep quiet. Depending on the immune system, factor of age, I hate to say some ethnicity issues, it is a very quick moving target. So quick. I don't know what we did in the early days with the Spanish flu back in 1918, 1919. There was no internet, there was no television at the time. Millions of people died. But now information is information spreading faster than the virus? Apparently the viruses are catching up and they're surpassing us. So new technologies have to be developed, which are very sensitive, cost effective. If it has to go to a little village in Africa, the cost should be very low. There's an incubation period, then you begin to see the viral infection occurring through the spike proteins. So we're concentrating on the spike proteins, as I said earlier. Serology testing to detect the immune response is very important. The diagnostic test for the COVID to diagnose an acute COVID infection. For serosurveillance of COVID infection, there are two ways of looking at it. A molecular test is used to detect genetic material of the virus present in respiratory samples. These tests use secretions, mainly as a swab. Swab is easier. Even in the case of children, swab is okay, except you don't need to prick using what we do for a glucose measurement, which is routinely done. That's diabetes. I'm not going to go into that right now. What is cross-reactivity? Cross-reactivity occurs when antibody is produced against the proteins. One type of CoV can occasionally bind weakly to proteins. So there is early onset of the infection, more advanced, and when it gets beyond that and we are not detected it, it could get serious. That's what is happening in approximately 9% of the cases. And people who don't have access to world-class infrastructure in healthcare, even in this country, in some far-off places in Alaska, or in New Mexico, or Kansas, or Idaho, or anywhere, this problem is very serious. Public health importance of testing. The testing is important to identify those who are infected so the patients can be isolated and onward, transmission prevented, and their contacts followed up. That is a very difficult thing. We are not able to do it at all. When will a COVID vaccine be ready? I'll come to the vaccine issue, is also related to the production of antibodies. Which antibody is the most potent? We don't know. We think that camel antibodies are not going to pan out. Alpaca is gone. Shark is gone. Maybe we'll have to go back to the bats themselves. If the bats produce it and they can host it, how do they host it? Maybe we'll have to go back to the wet market in Wuhan, which is a much detested place, even in China. They're closing down everything. Maybe we'll have to go to the bats. I don't know. We know enough of the biology of the bats at the moment. Comparing the traditional, I'll come to the vaccine completely. Okay, later. I'm going to do that. How do we again get out? Nope, we don't need this. This is something from Australia. Nope. This project is centered at the COVID Innovation Center in GB. We have a group in Germany, University of Potsdam. We are collaborating with them on the monoclonal antibodies. This is the Stanford group here. This is also Stanford. This is Berkeley. Then the University of Wisconsin School of Medicine, as well as the COVID Innovation Center right here is doing research. The entire group is MD, PhD. Now, as I told you, we have been able to get glimpses of what happens in the very early stage when the virus, the spike protein, enters the host cell. What happens? There are membrane proteins involved. Then it makes an incision and goes and takes over the cellular machinery of the host cell, aggressively takes it over. In this process, one single amino acid mutation, one residue mutation, as I said, D614, D stands for aspartic acid 614 going to glycine, is we thought is the most important one. First report of it came from Korea. Then it jumped into Malaysia, Vietnam, Myanmar, Bangladesh, India, and all that. Now we see many more coming in. Showing a schematic of this. This is a, first of all, It's a small device, very small device, about 2 centimeter by 3 centimeter. All it needs is the swab to be introduced or a very small, tiny nano droplet of the blood or saliva. In the case of children, it's a lot easier to work saliva. And that enters, and then it is divided into several aliquots or portions. Each one goes through a channel. One channel we detect the virus, second channel we detect the antibody, third also is for antibody. If antibodies are present, if the patient had been asymptomatic earlier, then the whole thing is coupled to a PC, and then we can begin to look at the signals that are coming out of it, and we can color code them too. In case you detect the virus, you'll get a red. In case you see the antibodies, you'll get a different color. Now, if you look at the human, there's a receptor binding domain, and this is the spike protein in which the mutations occur in various places, especially in the position 614, which is aspartic acid. This is a schematic surface how the antibody, the spike trimeric protein, is a trimeric protein. It's S1, S2, S3, an antibody bound to the cryptic epitope of the spike protein. These are the linkers to the graphene. This is surface, the graphene comes in here. And some of the early results indicate sensitivity is very high, which is very important. Rapid throughput. You can use a disposable system, which will be a lot simpler and reduce the cost. And these are all the experiments to demonstrate how the spike protein in our hybridoma-derived cell lines and patient convalescent plasma, we switch to plasma too, react. How do they neutralize it? These are all the neutralization data. I think you'll get too much technical if I go into these things. The virus recognition is very different from bacterial recognition. Antiviral agents are far more complex than antibacterial. The size of these bacteria and the virus also are very different. They're tiny particles. Right now we are waging a war with nano, sub-nano. It's not a huge enemy, but a silent enemy. Now, these are all some data from antigen-specific screening. I want to spend some time in this. This is how a device based on FRET and a graphene-based sensor will work. You have these are the various channels here. This is outlet, sample inlet, and here is one of these units here. What is happening here, we take the antibody, bind the antibody chemically to a single layer graphene surface. And that is again, the engineering part of it comes. I'm not an engineer, not a practicing physician. I cannot be all at one time. But today's world demands that you have an exposure to all the various. I'm now addressing to the students, if any are present, I'm sure there are. It is very important that you look upon science as one whole unit, not as compartments. So this is how these things look. The antibodies are attached to the graphene layer. And then this is a two-dimensional layer here. The electrodes are connected to the electrical connections. This is the whole thing is a plastic called the microfluidic encapsulation of that. And all this is entirely fabricated in the bioengineering at the University of California, Berkeley. Okay. And then the readout is purely electrical. It's a very small device. I would say at the same size as an oximeter we have. The difficulty of going to the hospitals, the risk involved in it is driving people to set up many things at home. So they should also become knowledgeable. How do you make less, privileged sections of a vast country like India aware of all this? That is where we need a lot more work, and it's not going to be easy. We are only going to cover some percentile, a double-digit percentile, I'm sure. The rest of the population is going to be left out. They have to be brought on stream. It's simply not for those who can afford it or who understand what is going on and will take timely action. Now, were you able to see this?
Yes, sir.
Okay, because I need some input from you too. Yeah, so far on the testing, is testing the end of the whole process? Absolutely not. After testing what? That is where the vaccine development comes in. Now, if you look into the vaccine story, there are many, many antibodies that are being considered. The co-vaccine, which actually came out of India, is still under clinical trial. There is no point in setting a deadline on this. Scientific research doesn't go precisely according to the wishes of people or the political mandate in a country. It was based on looking at the antigenic domains in the virus, the epitopes, and trying to use a limited extent of that information to create a vaccine. From what I understand from the vaccine groups, both at the National Institute of Vaccine and there are many institutions in Delhi, as well as the Amity University of Jaipur campus, they are still under clinical trials. We don't know whether they are going to work. But unfortunately, I do not see them among the major players in this country. Why is that? Number one, you need a lot more cooperation within India. You got a talent pool in different places. You need a Sardar Vallabhbhai Patel to be able to integrate all this, and that person is not going to be very popular. The way we look upon collaboration here is different. Every collaboration is a strength to us. They leverage me and I leverage them. We're happy to say that openly. And who leverages more depends on how well it is leveraged. Okay. In the Indian context, the groups are isolated. There need to be more dialogue between them. Currently, some of them are proprietary. At this moment, Moderna, Gilead, Pfizer, AstraZeneca, of which we cooperate with Moderna and AstraZeneca. We have signed an agreement with Moderna. Will it work? I can't bet. Maybe we should consult an astrologer whether it will work or not. Nothing has worked in the past. So the vaccine development is the next step, very important here. There's no timeline. There is one set of slides that I thought I had permission from them to display it. I'll be happy to go through it. I don't think there is enough time. You're already going to be just 1 hour. Not that 1 hour is set in granite, but people have other things to do. We have to value the time of everyone here. If you are interested, I can bring someone from Moderna to talk to all of you again, if you feel it will be worthwhile. Dr. Ilangovan. Hello? Can you hear me?
We can hear.
Yeah. We can set it up 2 weeks later or something like that. We can go through the vaccine development. People are asking this question, where is the vaccine? Has it been tested? Yeah. Okay, you can detect the virus. What do we do after the virus? The next step is a vaccine. Is there going to be a single vaccine? No. It's going to be multiple vaccines. Vaccines maybe have to be tailored to that person. This is asking for trouble. They're talking of less than 6 billion people in the world, each or a group of them requiring one vaccine, a second shot, primary, secondary shot. This will cost a huge amount of money. I will be happy to talk about it, provided I get the permission from Moderna to talk about it. I am not the representative of Moderna. I can tell you what they're trying to do, provided they let me do it and you are interested. Dr. Ilangovan.
Yes, sir.
It's your call. I'm Ramesh. We can talk 2 weeks later. I can bring in one more expert in this. It doesn't have to be me. I am not holding all the secrets in my hand. I'm not. We are one of the players. Okay.
Thank you, sir. Thank you.
Thank you. I hope I've given you some idea. I'm not sure how well it is. Just a moment. Next time I need to put them all in one single frame. Send it to you after getting the permission so that you can display it, Dr. Limon.
Okay, sir. No problem.
Okay. Very good. Thank you, Ramesh. Thank you all.
Ramesh,
I
sir.
appreciate this opportunity.
Finish.
It's nice having all of you with us. Okay. Thank you. Bye.
Sir, any questions? You would like to take 1 or 2 questions, sir, from the
I'll be
audience?
happy. I'll be happy to.
I would like to request the audience to make your questions in the chat box. I would greatly appreciate.
We have.
I'm sorry, it is a more specialized lectures with a lot of research background given by Professor Dr. Renu from Harvard University. I hope it will be a useful one. And if anyone really interested to make any questions, I can share the email and contact of Professor Renu.
I have one advice. Should you think of having one on vaccines, it's better to involve the group in India too.
Yes.
Maniti University, all India Institute of Medical Sciences, infectious disease. That way, there will be a balance between the American and Indian experts in this. Some work is going on, very interesting work, but it needs a lot more trial. There are some areas that I would like to avoid any path of collision with China. Number one, any political connotations of this, especially in the genetic diversity. Third, on the vaccines.
Okay, Dr. Renu, I have a question. In India, after this pandemic, most of the population, I mean, in semi-rural, rural areas, and probably semi-urban areas, they were all practicing natural herbal medicines, some vitamin C. This is the most common items they were used to take, and seems that they're recovering, right? Medically, I am not an expert to say.
Every morning here, there is a discussion on alternate medicines.
Great.
In fact, I'm myself taking some of them. Are they really producing effects or the placebo? I don't know. I have not seen any dramatic changes. Vitamin C has been around. Linus Pauling proposed vitamin C for infection. You know, he was a little bit eccentric in his days. Then Dr. Fauci thinks we need vitamin D3 or D6. Also one of the vitamin B12 or B6. So, and then there are natural remedies too. We should give them some credit. China and India has many traditional medicines. How do we know they don't work? There's no way of ruling it out. I think we must keep an open. I don't know they are harming the system. So if it works, it works. If it doesn't work, it doesn't work. Right? You take Patanjali Laboratories, there is some controversy going on, but leave aside the controversy. Is it really working? Then we should give them some credit, right? But one unfortunate thing, none of them, you can go to a drugstore in the United States to buy them. Whole Foods, Jeff Bezos has started selling some of them. So there's a continuous argument in China too. They claim there are many remedies, but I haven't seen them. So,
yes, sir. There is a question from Darshana. The question is there any issue for creating antigen rapid test against COVID-19?
that's what I've been talking. I've been telling you that this little graphene-based microfluidic sensor, 2 centimeter by 3 centimeters, when you put the swab into that, insert it, or like a lancet, as you do in the glucometer, it'll look for the virus and tell you is the virus there. It'll also tell you antibodies are there. If you had any Unfortunately, I had asymptomatic COVID, which I suspect I had, and Chinese never warned me. I was taken by the wining and dining and the banquets and the Chinese motai liquor going into my system at a rapid rate. Probably I lost my sense. They have a way of mesmerizing you. They're throwing money, enormous amount of money, unbelievable, attracting the Internal Revenue Service to my door sometimes. As India is not doing it.
Thank you.
That's a different subject. I'm not going to go into that right now. Yes, that is what I've been talking about.
For your,
Directly, yes.
thank you for your
Virus
extensive
itself.
lecture. And once again, thank you, Professor Renu. And I hope to see you soon in Massachusetts. And in case
Awesome. You're welcome here, but this place
welcome
is
back to
dangerous.
Bangalore.
This
And-
place is dangerous right now. Are you hearing me?
Yes, sir.
We cannot ensure your safety here. I have not gotten out of the house maybe a dozen times.
Right.
And every time I have to go, I think I'm ending up in the ICU.
Right.
So there's a real problem for all of us. It's simply one country alone. Underreporting is going on. This is what bothers me. I don't know. I believe the numbers may be very, very staggering eventually. Maybe some one single digit percentile of the entire globe. It might be, assuming it's about 5.8 billion or 6 billion. It's a very large number. We don't have the resources, no matter whether the country is rich or poor. What about poorer countries? How can they afford remdesivir? No way. And
Yes.
I'm in touch with the WHO also. I've been talking to WHO almost weekly. We have a CDC alert. The CDC alerts are so scary. Sometimes I feel like avoiding them. I don't know what they're going to say next. We don't
Right.
want to hear bad news, right? Okay, go ahead. We can talk on vaccines. I will bring a man from the epicenter of it, Moderna Pharmaceuticals. Okay, after the permission.
Thank you, sir. Thank you
Okay,
so much.
thank you all. I'm very grateful to
Thank
you.
Thank you so much for everyone
Yes, thank
participating.
you.
Thanks.